All right, so we're going to continue into chapter 5 now, talking about the primary structure of proteins, introduction to amino acids. Here's the 20 common amino acids and their 3-letter abbreviations, which I had already asked for you to memorize. Some of these are a little bit confusing. If you look at glutamine versus glutamic acid, glutamine is GLN, even though it starts with glue. Okay, so glutamine is GLN, glutamic acid is GLU. What's another one that's common? Which one? Lysine and?
Methionine.
Oh, methionine? Methionine is MET. Why is that?
I would say that's almonds.
No. I'm talking about one that you might screw up. Asparagine and aspartic acid, right? ASN and ASP. So those two. The others are pretty straightforward. I think the others are pretty easy to memorize. So just those few that are a little bit confusing. We're going to talk a little bit about the pKa's and the ionization of amino acids. Look at the occurrence in proteins. I was going down through there and I was like, which one has the highest occurrence in proteins? And glycine is, or not glycine, but alanine is pretty high at 8.7. And then you go down to leucine, and leucine's at 8.9. And we'll talk a little bit about those two.
I have a question.
Yes.
So, when you're transcribing DNA, I think, methionine is your start codon. So shouldn't every protein in theory have a methionine in it? Mm-hmm. But it only is 2.0.
The others require repetition on the same protein.
That's true. That makes sense. Okay.
Okay. All right. Fisher projections, going back to good old organic chemistry, okay? And remembering how to do Fisher projections and what those look like. So, anybody got their model kit?
No.
I do.
If you had said
You
something.
got your model kit with you?
It's in the lounge.
Oh, it's in the lounge.
Do you want me to
Okay.
get it?
Well, I won't have you run off like Caleb.
I did
She a track star.
I don't think you could do a Fisher projection on like 1 carbon. Everyone we looked at organic, there were always 2 carbons.
Yeah, you can do a Fisher projection on a single carbon. Yeah, absolutely.
Fisher projection.
Yeah. Well, I guess, yeah, it would be 2 carbons, but you have to think about, a chiral center, right? Is this a chiral center?
Yeah.
Yes, it is. Okay. So your vertical is what's behind the board, and your horizontal is what's in front of the board. And when you are doing R and S, what is the rule that I give you?
Counterclockwise. Hydrogen's in the front, you input it.
That's right. Okay. R is clockwise or right hand. Okay. If hydrogen's in front of the board, then you name it the opposite. Okay, which would be S. All right. If hydrogen's in back of the board, then you keep the same. You keep the same orientation. So, if it's clockwise and hydrogen's in the back, then it's R. If it's counterclockwise and hydrogen's in the back, then it's S. Anytime hydrogen's in the front, you will always do the opposite or name it the opposite. Okay?
Did you sign up?
Yeah. All right. On the left is L-alanine and on the right is D-alanine. And if you look at the hydrogens and the methyl group for alanine, because the methyl group is the side chain for alanine. Okay, the methyl group is here. It's in the front, and the hydrogen is here, and it's in the front. Okay, so everybody see that? All right. So L and D. The left hand is the L L enantiomer, which is the S. That is the natural isomer for nearly all amino acids, not all amino acids, okay? Also, not all L amino acids are the S enantiomer either, which is interesting. And the one on the right is the D enantiomer of alanine as well, which is the R.
Wait, so most of the time it'll be S, but there are instances where the L enantiomer is R. Is that what you're saying?
Is
Yes.
there a
Okay.
definitive way to know which is which?
Oh yeah, by actually doing the stereochemistry, okay? So remember the D and L, lever rotatory and dexter rotatory, remember that those were not defined by R and S.
They were defined by positive and negative.
Yes, they were defined by positive and negative based upon what?
Their ability to rotate polarized.
Their ability to rotate plane polarized light, that's right, in a polarimeter. Yep, very good, okay? Okay. Which of these is L-serine? Which of these is L-serine? Two of these are D and two of these are L. It's a little bit harder because, why? Why is this a little bit harder to do?
Well,
So remember, in this instance, L is the S and D is the R, okay? So what are our priorities? What are our priorities? Let's just take a look at the first one, okay? Which one's going to be number one priority?
The nitrogen.
The nitrogen, because it is the most electronegative element that's directly connected to the carbon. So nitrogen is number one. What's number two? Yeah, the carboxylate. The carboxylate is number two. And therefore, if this is 1, 2, 3, we're going to the
Right. So
right.
R, so that's
Hold on, and hydrogen is,
in the back.
and hydrogen's in the back.
how do we know hydrogen?
So it's R.
Oh, this is
Because
good.
it's in
How
the
do we know
plane.
hydrogen's in the back? Because we put it in the Fisher
Yeah.
projection. So now this is a little bit easier for you to see, and that's kind of what I was getting at.
But how would you know without the Fisher projection?
Because again, the ones that are vertical are in the back, and the ones that are horizontal are in the front.
Oh, okay.
Yep. Ones that are vertical are in the back, and the ones that are horizontal are in the front. So let's just go back to here, and I already helped you do the first one. Now I'm gonna let you guys individually do B, C, and D, and discuss it with a partner.
Vertical, yes. Vertical is in the back, horizontal. You're saying A is, so it's like originally clockwise with hydrogen. So it would be
R and S.
BBS.
R and S.
Oh yeah, or yeah.
B, would be, so it'd be R, it'd be S, because he has, you're going clockwise, but hydrogen.
That's what I was thinking.
It's just in the back, and it's going clockwise. Yeah. Because hydrogen's in the back and it's going counter, I'm pretty sure. Because hydrogen in the back is going counter.
I don't know what that is. I don't know what
This
it is.
one, D should be going to S, but then H is S.
Shouldn't amino always go first?
Yes, because hydrogen's in front, and it's originally
Crazy.
clockwise.
But how do you tell the hydrogen's going? Because
Like,
it's
I just
supposed to
was
be
like,
left, we stick with hydrogen left. They get to do it. That's how I do it. I just put hydrogen left, because that's how you determine whether
Right,
it's inverted or not.
right, because
Nitrogen, if you go
I gave it
left,
to you as
counterclockwise, right?
L&D
First two are
or
pretty much
S.
down.
It goes to the carboxylate
Right.
house. If you go right, it
So
goes to
going
the
back,
other
so
house.
going back.
And then just see
That makes
what
sense.
the
What we need
structure,
to-
what the
right.
function.
I wouldn't give
I
you
knew
these.
there was some.
That one was S,
I
right?
just wasn't sure.
So that one was R.
With hydrogen in the front?
Hydrogen was in the front.
Oh, okay, then,
Yeah,
yeah,
but
then
it's
it's-
probably not going to test you on it, so. Not in that light, but we're looking at this. We're looking at this right now from the D and the L perspective. Okay, all right, keep going. If you're still arguing or debating it. No, it's not a new one yet. No. Raise your hand if you're still working on it. Okay, all right, this is good. What do you think, Caleb, Boulding? What do you think?
So which one?
What?
What was the question? I was just designating configurations. What was the question?
Oh, okay, so which of these are L serine and which of these are D serine? What do you think?
So, L is S.
Yes.
Okay, so,
Hold on, Caleb clearly didn't discuss it with
no,
you.
no, we did. I
Okay.
said right.
Oh, okay.
She was recording it, so.
She was recording.
Yeah, so A is D and D is D, right?
Okay,
And then vice versa, B and C are.
and then B and C
Are L.
are the L's.
Yeah.
Everybody get that? Yes? All right, good. That is the case, okay? So, we determined that this one was which one?
R.
Okay, so let's do it again. So 1, 2, 3, 4, we're going to the right, hydrogen's in the back.
So it's R.
So it is R, okay? Let's go here. 1, 2, 3, 4, to the right, hydrogen's in the back. So this one is
You mean
the,
the front?
You mean the front?
sorry, hydrogen's in the front, so this must be the
S.
S. So even though we're counting clockwise, hydrogen's in the front here. Okay, so you name it the opposite. All right, good. Here we go here. 1, 2, 3, back to 4. Okay, so even though hydrogen's in the way, we just bypass it, right? So you go 1, 2, 3, back around to 4. We're going counterclockwise. Hydrogen's in the back, so it is S. And here we're going to bypass it again. 1, 2, 3, back to 4. Counterclockwise, hydrogen's in the front. We name it the opposite. Therefore, this one is the R. Okay. All right.
Okay, I have a question. So how do you know definitively when you're looking at these if the R is D or if
We
D
just
is
talked
D?
about this, and that is that I'm not giving you the exception.
Okay.
Yeah.
So anytime.
Well, no,
you
but
have-
I mean, like, when we sit down to like learn them, how do we know which one is the exception?
So,
Does that make sense?
see if you can find it. See if you can find which one is the exception. Okay?
Well, there's only one amino acid that's the exception.
I think there's only one, maybe 2 amino acids that's the exception.
Okay.
I can't remember, but I know that there is an exception. Okay? All right. Here's a different perspective. This is what you were more accustomed to in organic chemistry, okay? This probably feels a little bit better when you look at this slide, right? When you draw a chiral center, you will always have 2 bonds in the plane, one in front, one behind. Always on a chiral center, all the time, okay? Except when it's in the Fisher projection, okay? When it's in the Fisher projection, you don't get that, okay? All right. Which one is this one? I know it's A, you probably may have remembered
R.
it.
S.
It's R, it's S, which one?
R.
This is
It's R.
good, this is really good.
R.
Okay. 1, 2, 3, back around to 4.
Hydrogen in the front.
Hydrogen's in the front, so it must be the R. Good.
All the OHs on the central carbon.
Good. What pH is this at right now?
Well, okay, it's,
What has plus A like?
What? It's protonated.
is it is it neutral?
Is it close to neutral?
Well,
Yes.
it's been protonated and deprotonated.
Okay, now, here's the thing. This would be where? Okay,
What do you mean?
what'd you come up with, Devin?
I think it's 2.2,
You think it's at 2.2,
or it's at 9.2.
or is it 9.2?
Those are two very different things.
Okay, let's think about this, What's the pKa of an alcohol for those of you who are in organic chemistry right now? Organic chemistry 2, pKa, typical pKa of an alcohol. 15 to what? 16 to 18, okay? Is there an alcohol in this one?
Yes.
It is, okay. What's pKa of a carboxylic acid? Yeah, really pretty acidic, right? Okay, like 2 or 3. Oh, but this one's a little bit different, okay? That's the conjugate base of it.
Forgot about the alcohol.
I believe this is where it's at its isoelectric point.
That's what I was thinking.
I believe it is, okay? So I think, Devin, it would be between those two numbers, which would be closer to physiological pH. Okay. All right. Anyway, that's besides the point. Let's digress. All right. Here are the amino acids that have non-polar aliphatic side chains. We have glycine, we have alanine, valine, leucine, and isoleucine. Which one of these do you think is the most water soluble, and why? Which one of these do you think is the most water soluble, and why? Why?
I think it's glycine.
There's less carbon.
That's okay.
I think it's glycine.
Yeah,
Glycine
all those have
on the top and alanine, okay? So why?
Because they don't have the carbon chains.
Less carbon chains, therefore probably more solubility in water, okay? I have dealt with these before, and that is the truth, okay? Yep. Glycine, what's really interesting is it doesn't really have that great of a classification. They've just kind of lumped it in here, okay? Glycine's actually found more on the surface of proteins than it is anywhere else. And I think that may be why, because it is more water soluble and it's found on the surface of proteins, therefore giving it a less non-polar surface, okay? But also the book talks about glycine's ability to form quick turns or tight turns in the structure, and therefore that gives it the ability to form more compact secondary structures, okay? So the peptides. Alanine, valine, especially leucine and isoleucine, these down here at the bottom, are actually more found in the cores of proteins. Why? They're more hydrophobic. Yeah. And therefore, being on the inside, then they're more shielded from water. Yeah?
I know like it's both polar and non-polar region, but aren't technically those still water soluble? Like, isn't there enough oxygen to have carbons to still make them water soluble?
Miscible. They are, and it also depends on their charged state too, right? Yeah, okay. Any questions on those? All right, what's the 3-letter acronyms? Glycine? Yep. Alanine?
ALA,
Yep. Valine?
BEAL,
Yep. Leucine?
LEU,
And isoleucine?
ISO.
Nope, not iso. Try it again.
Is it ILE?
It is IL.
U.
Is it ILE or ILU? It is ILE, I think. Yep, I do believe it is. Yep. Okay, good. ILE. Here are the amino acids with non-polar aromatic side chains. So these is the most non-polar. So these is the most non-polar.
Tryptophan?
Are you sure?
Well,
I'm not sure.
maybe not. It has more carbon.
Which of these is the most polar? Tyrosine, okay, but it's still considered a non-polar side chain because phenol is not really considered that polar, okay?
Does the nitrogen and tryptophan cyclopentane make it slightly more polar than phenylalanine?
And why would it?
Because of the electronegativity and the induction of the electrons?
The electronegativity and the possibility of hydrogen bonding interactions. That's right, okay, so phenylalanine is the most polar of these aromatic side chains. There is a disease that is associated with the intake of phenylalanine. Do you know what it is? It leads to buildup of phenylalanine in the body, which can eventually lead to mental retardation, and every baby in America is tested for it at birth.
Phenylketonuria?
Yes,
Yeah.
phenylketonuria, or PKU disease. PKU babies or patients suffering with this have to be on special diets to regulate the intake of protein. I had a friend who had both of his daughters were twins, and they both had PKU when they came out. If you see a parent at McDonald's with a scale weighing out French fries and weighing out food, it may seem very odd and strange and even a bit, controlling and manipulative or mean. And these are parents with children who have PKU because they actually have to keep on track of the exact amounts of their dietary intake. So they will actually weigh food and give it to their kids. They were really looked and frowned upon, made fun of, and they had to explain, to people, look, this is why we do this. But some people just, didn't understand. Yeah, Riley?
People used to get mad at my parents for thinking they had me on a diet because they would weigh out my food in restaurants and like stuff like that. And I was like small for my age, like really small for my age, and people would get really mad at my parents.
Yeah.
I thought that they were like starving me.
Yeah, it's really kind of wild, the types of, things that people come up with or think about. Yeah. Anyway, so there is currently no cure for PKU, and there is research going on out there. I used to do a race out in Colorado, a cycling race to raise money for PKU research through Denver Children's Hospital. I did that for 2 or 3 summers in a row. Okay. Amino acids with nonpolar aromatic side chains. This is tyrosine. Tyrosine actually ionizes at a pretty high pH, and its pKa is at 10.0, which you should know that from organic chemistry because phenols have a pKa of around 10. That's right, okay? Now, really close to that. Tryptophan and tyrosine both absorb at about 278, 280 nanometers, somewhere in there. And that's actually why you can, you can use UV Vis as a tool for looking at protein concentrations. Cysteine's a really interesting one. Cysteine has a thiol side chain. It ionizes lower than that, pKa of about 8.3. What do you know about cysteine? What do you know about cysteine? Actually, I take that back. Let's look at cysteine, and let's look at its conjugate. Actually, its conjugate's on the board. Look at serine. Serine versus cysteine, what's the difference? Yeah, it has a thiol. Which one's the better nucleophile? They both will act as nucleophiles in the body, as a matter of fact. Which one will be the best? The thiol. Why?
Because sulfur is really electronegative.
Okay, sulfur is really electronegative. Oxygen's more electronegative. But there's something else about the sulfur.
Is it the extended octet?
You guys,
For a better nucleophile, wouldn't it be oxygen because it's more localized?
Oh,
Whereas
you'd
sulfur-
think.
whereas-
You'd think, but it's not. It's not.
is it because that can be deprotonated more easily?
Nope.
Is it because of the electron density?
She's getting there. She's really
Is it because
close.
electrons are more spaced out than oxygen?
It is the atomic radii. Yeah, it is the atomic radii of sulfur being a little bit larger that actually makes it a better nucleophile.
I'm very confused. That's what I'm saying. That is more delocalized.
My next guess was done.
So wouldn't that make it a worse?
But they're more available in terms
But that
of.
one doesn't.
Yep, it's a better nucleophile. Yep.
Then why is it like, okay, when you're talking on a bigger scale on a molecule, then why is more localization make it a better nucleophile?
It's the availability of the electrons. So the electrons are further away from the positive charge nucleus, right? So if they're further away from the positive charge nucleus, then they are less attracted to the nucleus and they are more prone for nucleophilic attack.
Wait, okay, that's also kind of confuses me a little bit. Like I kind of understand it, but at the same time, why is iodine not? Iodine is a good nucleophile, but it's not as good as like bromine or chlorine.
Depends on the conditions, actually. Yeah, depends on the conditions. Depends on the solvent, too. Yeah, we can talk about that in organic.
Because we don't use.
We can talk about that in organic. Yeah.
Do you know the pKa values of each of these?
No, you do not.
Also, you asked what we know about it.
Uh-huh.
It forms disulfide bridges.
That's good. Okay, it does form disulfide bridges, but why and where does it form those,
Well,
Caleb?
it's the sulfur molecule, the sulfur atoms, right?
Okay, yes, of course it forms it at the sulfur atoms, but
Within the
Okay.
tertiary structure and quaternary structure.
yeah. So that enables proteins to have greater stability when it comes to their tertiary and quaternary structures. That's right. Okay. This happens through a process of oxidation. Okay. So when you have cysteine and you oxidize cysteine, then you end up with the disulfide bridges, and then you can reduce it, and you can make cysteine back in its reduced form. Yep.
So I think there's another amino acid that has selenium instead of sulfur. Does it form selenium bridges?
You know what? I don't know.
Diselenium.
I do not know if it forms selenium bridges. That's very, very fascinating.
Which one is it?
Yeah, I don't know. That's a great question.
Because
I wonder if it's ever been observed.
it's selenocysteine.
Yes.
Sabo is selenium.
Yeah, selenocysteine, yeah.
This might be a dumb question,
Yep,
like I
it is.
probably
There are no
done this
dumb
a long
questions.
time ago.
There are no dumb
Formation of diselenium
So,
bridges
like, you
approaches
see
by
it on so
incorporation
many
of
different ways, like, so many
selenocytals.
times. So
So it
why
depends on
in this case we're supposed to
Psilocybin
hold on
bridges.
to like other bonds of each other?
I don't know if it
Like,
happens
how do you
naturally.
know?
So it depends on the form.
I think it just, it's the oxidation state, you know? I do think it's all about the oxidation state. I don't think I don't think there are many instances where you see sulfur in a +6 oxidation state in nature, maybe, or at least in an organism. I'm not certain. Not certain. Now phosphate, right? But I'm wondering about sulfate. Like, do we see sulfate that often in natural organisms? I don't know. That's a good question.
Diacylonide bridges confirmed.
What's that?
Diacylonide bridges are confirmed.
Are they?
Yeah,
Really?
it's selenocysteine residues.
So in selenocysteine residue, so you can get a diselenium, diselenide bond.
It just depends on the conditions.
Very fascinating.
I can't see the full article though.
Yeah, well, selenocysteine, though that conjugate is pretty rare, okay? I don't know the full story behind it. I did actually research it last night and read about it a little bit. But I just come back to it. Why selenium, you know? I don't know. Let's go look at it. There it is. There's selenocysteine, okay? And there's pyrrolysine, okay? Pyrrolysine I think is even more rare. But why selenium? I mean, I know we make use of copper and nickel and zinc and those metals in our diet, right? Selenium is actually important in our diet as well. Not a dietary, a dietitian or dietary major, whatever. The actual element itself, though, why selenium? Because it's similar to sulfur. Selenium's a non-metal. It is the lowest non-metal, I think, that we have up there, except maybe, I don't know, except maybe iodine, okay? But I often wonder why selenium? It's kind of fascinating.
I mean, it's chemically similar to oxygen and sulfur, so it makes sense they would bond.
Yeah,
But then again, logic, so it's one.
it is, but it's also more metal-like, you know? So, yeah, I don't know. And it's a lot heavier too, right? I mean, didn't we determine that arsenic was also, Caleb, you looked that up, that arsenic was actually beneficial in certain circumstances?
I think someone else mentioned, arsenic. I think I looked up the one about one of the other ones.
I know I think I looked up something along those lines, and I think it has a lot to do with prokaryotes, like the extremophiles maybe.
Oh, wait, I remember, yeah, I think she brought that up, and I was looking at the one about tungsten, how certain prokaryotes actually use tungsten in a similar acid.
Yeah, and I mean, you look at tungsten's heavier, I mean, it's really heavy compared to where we're at in the non-metals.
Just a little bit.
And we talked a little bit about lanthanum, remember the lanthanides? We talked about how bacteria they found utilize lanthanum, and that's pretty heavy as well.
I don't know what it's used for exactly, but arsenic, so I've been reading up on schizophrenia lately, and arsenic is, okay, so there's an arsenic pathway in the body, in the schizophrenia, that gene is damaged, for like, to deal with the arsenic in the brain, and I don't even know how that works, but apparently arsenic is in the brain.
Oh, okay, interesting. I think it'd be good to look it up. Yeah? I know that lithium's used to treat people with bipolar disorder, and so that's another metal out there. All right, we're off topic. Let's get back. Okay, here's some typical ranges for pKa values for ionizable groups and proteins. And this should be pretty straightforward. We should know that the carboxylic acid group is that lowest range, right? Because of the most acidic, right? So carboxylic acids are the most acidic ones. Side chain carboxylates a little bit higher. Imidazole, which is gonna be the next slide, looking at histidine, is closer to the physiological pH, 6.5 to 7.4 for its pKa, and then you just keep going on up through there. Arginine, look at that, like 12. It's pretty high. All right, so let's take a look at histidine here. What do you notice, kind of right off the bat? What do you notice? Yeah, a lot of hydrogens that it can gain or lose and ionize. So you have pKa1, pKa2, pKa3. You got 3 different pKa's here. Its isoelectric point, again, 7.6. You've got that range for physiological pH right in here. And you can see you've got the positive amine group and the negative carboxylate. And this is why I was telling you about serine previously being close to its isoelectric point as a result of these 2 charges that exist, okay? And that's how you know that you're close to that isoelectric point just by looking at the structure of the molecule. So anybody have any questions on that?
How similar is histidine to histamine?
I don't think it's very similar. No, I don't think it's very similar. I don't know. I could be wrong. It's been a while since I've looked at histamine in its structure. Somebody want to look it up and see how similar it is? Histamine versus histidine.
Because I was curious, because like histamine is blood clotting, so it's really from the bloodstream.
Mm-hmm.
Histidine has what appears to be a carboxylic acid attached to it.
You mean histamine?
No, it is histidine.
This is histidine.
The Google's blank.
Okay,
Histamine.
histamine is formed by the decarboxylation of histidine.
Oh,
Histidine has
it's
a
formed by
carbon
the decarboxylation of this.
Mhm.
Huh, there you go. Okay, so I'm assuming that is the source then of histamine is through a decarboxylation of this amino acid. Yeah? Excuse me. All right. Here are 4 post-translationally modified amino acids that I have never studied before. Okay, I do not ask that you remember these, but they are interesting. Phosphoserine, so you got a phosphate off of the serine group. 4-hydroxyproline. What is that? Is that epsilon?
In epsilon.
In epsilon, acetyl,
And that one's gamma.
is it gamma?
Gamma carbox.
Oh, no, I'm talking about the acetylysine. I could not pronounce that for the life of me today. All right, and then the gamma carboxyglutamate. Okay. They are the rare amino acids that we talked about. Just actually, there was something else the book mentioned. We'll go back and look at this. I can't remember what it was. Oh, the pyrrolysine is restricted to a few archaea and eubacteria. So it is even more rare. It's even more rare. What else? I would be interested to know how selenocysteine is encoded. I'd be really interested to see how it's actually encoded. I'll have to look that one up. Where would it be encoded at? I mean, if it's genetic if it's inputted into genes, it has to be encoded for, right? Yeah, right? Okay, let's talk about peptides now. What happens to amino acids? What happens to an amino acid in order to form a peptide?
Condensation reaction.
Okay, so a condensation reaction, and where does that reaction occur at?
Between nitrogen and
Between the nitrogen and the
carboxylic acid.
carboxylate or the carboxylic acid group. Okay, so a condensation reaction that liberates water. Okay, so here's an example of a peptide. Let's put together the N-terminus and the C-terminus. The N-terminus is the amine end, the C-terminus is over on the carboxylate end. Can we synthesize these in the lab?
Probably, yeah.
Yes, we can. Yes, we can. Okay. What else can we do? How else can we synthesize them? Inside an organism. Okay. How would you do that?
Culture bacteria include a gene that encodes whatever you want to make into the bacteria and let them make it.
That's right. Genetically modified organisms. Okay. Recombinant technology. Right. So this is, matter of fact, how my son's medicine is made through E. coli, and then it's expressed through that. So, interestingly enough, the formation of peptides is thermodynamically unfavored. How does it happen? If it's thermodynamically unfavored? Even better question. How did it happen in the primordial soup if it is thermodynamically unfavored?
You got struck by lightning.
It increases the entropy. Is that right? Because if it's
Okay.
not thermodynamically favored, it has to do something to offset that.
Okay, now, here's the thing. How does it happen in our bodies?
Catalyst.
Yeah, enzymes. That's exactly right. Okay, so enzymes overcome this barrier. Okay, and of course, me talking about primordial soup, that's just me snickering. Okay, all right. We've never been able to get anything to come out of that. All right. What about enzymes? Why do enzymes do this?
Because they lower the energy of activation needed.
That's right. That's right. They lower that activation energy needed for this to happen. And they choose a different pathway, right? And that pathway is altered, and that's how all catalysts work, as a matter of fact. They change the chemical pathway and get around that barrier. Yeah, Caleb?
I was gonna ask what you meant by pathway. I didn't know if you meant like physically in the cell or
No, chemically, mechanistically. Yeah, that's right. Okay. Here's some sequence specificities of some enzymes and where they cleave and how they cleave, and they're very, very specific in what they do. And that is it for today. I'm not going to go into that. We'll come back to that on Monday. Talk about that. Please discuss in the forum the procedure, and then I would like to gather materials and do that. If not, next class period, if you guys are able to determine which one you want to use, that can just be something fun that we do. So on the midterm, chapters 1 through 5, remember chapter 3 is only that small bit on ATP. Yep, and I will send out a study guide as soon as possible. I posted the chapter 4 slides. I will post some of the chapter 5 slides. Yeah, not all of chapter 5 is going to be on the